Good evening to all of you. We are back again with another exciting session. I, Ajay Jha, on behalf of ACS India team, welcome you to today's ACS Science Talk. Today, we are pleased to have Professor Vishal Rai with us. Professor Rai is currently Associate Professor at the Department of Chemistry, Indian Institute of Science, Education and Research, Bhopal. He obtained his PhD from the Indian Institute of Technology, Bombay. Thereafter, he was a postdoctoral fellow at the University of Toronto, Canada. Professor Rai's research is focused on exploring chemical methods for precision engineering of proteins, which he'll be talking today. He has a long list of awards and accolades to his credit. To mention a few: Swart Jayanti Fellowship by the Department of Science and Technology, Department of Atomic Energy BRNS Young Scientist Research Award, Ramanujan Fellowship, Young Scientist Award by RSC WIS, and many more. So before Professor Rai starts, couple of instructions for the audiences. We request the audiences to put their questions into the Q&A tab on their screen. Kindly avoid putting questions into the chat box. And in case of any internet issue, please stay with us. Now I would like to invite Professor Rai to deliver his talk. Over to Professor Rai, please.
So thank you, Dr. Jha, for kind words of introduction. At the onset, let me also thank the ACS team for creating this wonderful platform and giving me this opportunity to share some of our research activities with all of you. So our group at IISA Bhopal is developing chemical technologies for precision engineering of proteins. And while we are doing that, we are also looking forward to see how we can create unique opportunities in the field of biology and medicine. Now, before I get into the details, let me tell you why such technologies are absolutely essential in a broader perspective. So the basic knowledge, So the basic knowledge of reactivity and diverse aspects of selectivity with proteins provides a great opportunity to contribute to the market of biophysical and biochemical tools. Now, the fact that this segment is doing pretty well, even with the non-selective kits, tells a lot about the potential opportunities that lies ahead in front of the selective technologies. But what really excites us is the potential to contribute towards the directed cancer chemotherapeutics by synthesis of homogeneous antibody-drug conjugates, or in short, ADCs. Now, it provides a state-of-the-art technology, and as the name suggests, it's a combination of a monoclonal antibody that is connected to a drug or a toxin molecule through a linker using covalent bonds. So when these ADCs are given to the patient, the job of the antibody is to go and selectively bind the antigen that is overexpressed in the tumor cells, but not in the normal cells. So that's the basis of differentiation of the two. And then this whole ADC antigen complex gets internalized, and over a period of time, the lysosomal degradation releases the toxins, which eventually leads to the death of the cells or apoptosis. So based on this platform, there have been a few drugs that got approved recently through FDA and are available in market. And they have one thing in common, that they are sold as heterogeneous mixtures where 0 to n number of toxin or drug molecules would be attached to one molecule of the antibody. Now, this heterogeneity is definitely detrimental to the pharmacokinetics and brings in batch-to-batch variability. But not much can be done to solve this problem due to the lack of enabling technologies. Now, we have been working on some of these challenges in the past few years, and I'll be discussing about how some of those can be solved in the latter part of my talk. But while we are working with protein-based therapeutics, we also realize that this segment of therapeutics would not be accessible to a huge population across the globe simply due to the cost factors. So while we are working with homogeneous ADCs, in the long run, we want to see if we can selectively target a specific site of a specific protein within a human body. Because if we are successful, it could provide a disruptive platform for precision therapeutics because then small molecules can provide the specificity that has so far been possible only with the biologics, and that would be amazing. But before we can work with the complex systems such as humans or tissues or even live cells, we would first need the knowledge and methods that can allow us to precisely engineer the isolated proteins. And once we have that, then we can systematically translate them towards the more complex systems. Now, at this point, many of you might have a question that do we not already have drugs that are highly specific in nature? So let's take a look at one example to elaborate on this point. Kinase inhibitors have been involved in multiple critical signaling pathways in the human body. So it's not a surprise that they are very well investigated over past few decades, and they have led to multiple FDA approved drugs. But if you look into the most specific protein kinase inhibitors that are out there, they still go for at least 30 to 100 identified or known targets. And then there are many unknown ones beyond that. And this is the reason why these drugs have multiple off-target activities and leads to the side effects in the patients who administer them. Now, we are trying to see if we can address some of these challenges in the long run. So the first step in this direction was to see if it will be possible to even develop chemical methods that can render site-selective modification of native or engineered proteins. So in this perspective, we started by looking at protein as a multifunctional organic molecule, which is full of nucleophilic residues. So for covalent bond formation, we will typically add an electrophile in the reaction mixture containing a protein, and then all the nucleophiles are going to compete for it. So for precision modification, at first, the electrophile will have to figure out how to differentiate one type of functional group from all the other nucleophilic groups. And once we have such a chemoselective electrophile, you would notice that, let's say, if this electrophile is chemoselective for primary amines, then you can see that there are going to be multiple copies of the same functional group. So this electrophile will have to then figure out how to differentiate one particular site from all the other copies that are present in there. And that is what I'm going to term as site selectivity in the rest of my talk. Now, so for precise single site modification, we would need an electrophile to exhibit both chemoselectivity and site selectivity. Now, let's say this is the most reactive and solvent accessible amine, and it undergoes preferential modification with an electrophile. But what if we want to attach a probe at a different amine, let's say this one or this one? Is it even possible to have such control over selectivity? Now, this is what I'm going to refer to as modularity in the latter part of my talk. So now with this background, we asked a very basic question that are chemical methods capable of identifying the reactivity hotspots that are present in the protein? And after our initial investigations, we learned that the N-terminus alpha-amine, which in this case is over here, they serve as a preferential reaction center in multiple proteins and can be labeled with a high precision with an appropriately designed electrophile. So with this in hand, we now wanted to see if chemical methods can differentiate the reactivity of a residue from its multiple copies. And we were delighted to learn that not only this is possible, we can regulate the chemoselectivity and site selectivity with absolute precision for modification of even highly challenging residues such as lysine or histidine. Besides, we can apply these methods to structurally diverse set of proteins, install different probes of interest, and control the type of reaction or type of bonds that are involved in the bioconjugation. Now, one of the things that is worth noting at this point is that all these methods that I talked were limited to targeting reactivity hotspots in the protein that do offer high reactivity and solvent accessibility anyway. And at this stage, we really wanted to see if we can go beyond the reactivity order defined by the protein to develop modular technologies. The potential opportunities that can be accessed with such methods was a great driving force for all of us to investigate this. But such a technology also required us to address a combination of challenges. And we came up with a theory to do that, and that has been practiced in our laboratory for quite some time now. And the idea is that if we can disintegrate various challenges with the help of, let's say, a multi-step chemical transformation, then this disintegration allows us to find these solutions in a much better and controlled way, and then we can integrate the knowledge that comes out of it together to come up with the final solution. So just to give you a perspective of how this DIN theory works, if we add an electrophile to the protein, let's say in this case, for the N-terminus modification that I discussed, that single step had to take care of both chemoselectivity and site selectivity. And that was a bit tricky. But in another case, what you can do is that you treat the protein with aldehyde that can give a chemoselective and reversible reaction with all the amines that are present out there and generate electrophilic intermediate on the surface of the protein. So the reactivity landscape of the protein is redefined. The first step is chemoselective. So now in the subsequent step, if we are adding a nucleophile to make it react with one of the imine, it only had to worry about the site selectivity. And that is interesting because now it is far more convenient to develop new methods with this approach because you know what to tune when one or the other factor goes wrong. So with this, we hypothesize that what if we can take this electrophilic intermediate and generate another reactive intermediate with it? Because if we can, then this additional step might allow us to explore new dimensions of selectivity. So let me give you an example that allowed us to validate this hypothesis. So we took glycine and treated it with a very special aldehyde that has hydrogen bond acceptor in the proximity. And under physiological condition, it ends up giving us amino alcohol products. So the way it works is that the amine will react with the aldehyde first, generate an imine, and the hydrogen bond acceptor in the proximity promotes the generation of a nucleophilic enol type of intermediate over here. And this in turn reacts with another molecule of aldehyde to generate amino alcohol, which upon subsequent hydrolysis would release the amino alcohol as a product. Now, what is even more exciting is that if this R1 is not hydrogen and if it's anything else, then the conformation required for the hydrogen bond acceptor to promote the generation of this nucleophilic intermediate is not even adopted. So this is not there. In that case, there is no reaction. And that's exactly why this reaction would work only with glycine, and it will do a great job. But then if you have any other amino acid that's substituted, you will just have no conversion. So that was a remarkable residue specificity towards the simplest amino acid out there. And we took this technology forward and demonstrated that we can apply it to various proteins, and it just works pretty smoothly with all of them. And the fact that you can express recombinant proteins with N-terminus glycine pretty conveniently, such as sumo in this case, it made us far more general methodology to apply it for expressing proteins and tagging them with high specificity. Besides, you can club it with late stage tagging platform that can also allow you purification. So for that purpose, we had to redesign the GlyTag reagent a little bit. We made this a symmetric molecule. And now if you take a protein like insulin that has two chains, one would have N-terminus phenylalanine and the other one would have N-terminus glycine. So if you treat insulin with such a GlyTag reagent and let the reaction go for a while, and then you can take this mixture and along with acyl hydroside functionalized resin, you can conveniently capture this aldehyde through hydrazone formation for single site immobilization of this insulin. And you can also recover the unreacted insulin with very high efficiency. Now, this can be stored pretty conveniently, but what you can also do is take it into 3 different tubes and treat it with the hydroxylamine derivative of various probes of your interest, which will allow you to do transoximization and install the probes, and the spin centrifugation would release you and give you the analytically pure single site tag proteins. And that's really something that works very efficiently. So we took coumarin tagged insulin from this table and demonstrated that the binding of this modified insulin with the insulin receptor or its downstream signaling pathway remains largely unperturbed. Now, with this results in hand, we really wanted to see if we can apply this technology to more complex systems where more than one proteins are present. So to begin with, we took a mixture of 7 proteins, and one of them had N-terminus residue as glycine, whereas the other 6 proteins had other different residues. And upon subjecting it to the GlyTag reagent, we found that the chemistry still is very specific for targeting the N-terminus glycine of this single protein, which is insulin in this case, without touching anything else. So we were all set to see if we can translate this forward to a complex mixture of proteins coming from cell lysate. And that's exactly what we did. We took cell lysate where we expressed a recombinant protein, which is sumo in this particular example. And upon treating it with the GlyTag reagent, we were able to tag it with very high specificity and install the probe of our interest in this particular sequence of reactions. So with this in hand, we really wanted to see if we can somehow figure out how to learn to dissociate this carbon-carbon bond, because this was really important for many of our future applications where we want to translate this chemistry. So in a nutshell, dissociation of this carbon-carbon bond was not that straightforward, because in our initial investigations, when we treated it with various organic bases, really nothing happened. It was pretty stable in all those conditions. So after a lot of screening that I'm not going to discuss, what we found eventually is that we can treat it with a reagent like PLP, where this aldehyde would react with this amine to generate imine, and this extended polarized conjugation polarizes this electron density of carbon-carbon bonds substantially enough so that even under very mild conditions, you can dissociate it without any problem. And that releases the GlyTag reagent, gives you the adduct, which upon subsequent hydrolysis will give you the PLP back, and along with that, your native insulin is back. So now we had access to GlyTag chemistry and retroGlyTag chemistry. And we were all set to see if we can translate it further to develop a single amino acid or glycine based covalent tag for metal-free protein purification. And we found that it really works well. So we developed a GlyTag purification resin, and you take the mixture of protein from a cell lysate, mix it along with the GlyTag purification resin, and after a while, you wash off everything that is not immobilized, and you are all set to treat it with PLP for the carbon-carbon dissociation that I talked about, which releases the pure protein. And this is something that really works smoothly, and you really don't have any metal involved, so it's a metal-free protein purification protocol. Okay, so with this, let's move on to the final question that I would like to discuss today. That can we have a modular technology that would allow us to precisely label native proteins and hopefully at more than one sites? Now, while we were looking at this question, we knew that if we keep targeting a single residue at a given point of time, we will not be able to go beyond the reactivity hotspots that are defined by the protein itself. So we came up with an idea that what if we target two residues simultaneously? So in this model protein fragment, I have highlighted a lysine residue along with a histidine residue. So the idea was that if we can design a reagent that has two functional groups, FK and FH, such that the FK can give me a very rapid and reversible reaction that is chemoselective for lysine, whereas FH can give me a relatively slow reaction that's irreversible and chemoselective for histidine. Then if I have such a reagent that can be mixed to the protein or mixture of proteins, then the first thing that the FK will do is that it will just go and react with all the solvent accessible primary amines. And then it gives you an opportunity to control or regulate the micro concentration of FH near a particular histidine residue, simply by the way you design the spacer that connects the two functional groups. Now you should notice that this desired pathway is an intramolecular one. And any background reaction, if it has to happen, it will have to traverse through an intermolecular pathway. So that's something that plays in our favor. So we took this forward, and I'll show you one example of a model protein where things work pretty well. So in this case of myoglobin, we can label or modify histidine 36 or histidine 81 in two completely different domains without touching any other residue, simply by the way we position FK and FH in three-dimensional space with respect to each other. And not only it works well with myoglobin, we can translate it conveniently to a structurally diverse set of proteins, and it just does a great job with most of the proteins that we have tried so far. And once the job of FK or linchpin is done, it's all available for you to use it for late stage modification. So we can install a variety of biophysical and imaging probes through oxime formation, or even better, we can take it in the same way as the previous chemistry. We can take it forward, mix it with the acyl hydroside functionalized resin, so we can remove the unreacted protein, and now we have pure ordered immobilized protein on the resin ready for installation of whatever probe of interest that needs to be installed, and that release of the protein gives us analytically pure protein. So now we were all set to see how LDM platform is going to work for the synthesis of antibody-drug conjugates. But before getting into it, let's have a quick recap of some of the challenges that needs to be addressed in this perspective. So I'll show you a quick video to help you with that. Just a minute. Okay, so as we discussed, the antibody-drug conjugates are composed of three main components. The toxin or the drug is the first one, and that is what is highlighted over here. The monoclonal antibody, which in this case is trastuzumab, is the second component. And then you will need a linker that can connect the two of them, the toxin and the antibody. Now that also leads to the first question: how do we control the homogeneity of bioconjugation under such scenario? Now, once that is solved and the ADCs are injected into the bloodstream, the bond strength that connects the toxin to the antibody becomes an important factor. Because if the toxin is released before the ADC reaches the tumor tissue, then these toxins are going to have off-target activities and they will go to the normal cells and will have damaging effects in these cases as well. So the chemistry and the bond type becomes pretty important in this regard. Now, eventually the ADC will reach tumor cells. And once they go and bind to the antigen or might not bind, that's the step where they might or might not kill the tumor cells. So this is where the control over the conjugation site becomes critical to ensure that the ADC antigen complex does get internalized systematically without deviating too much from the actual pathways. And once that happens, eventually the lysosomal degradation would release the toxins. And as we discussed earlier, these toxins are going to be responsible for killing the tumor cells. And that's what you are observing right now in the video. Now, what you would notice, or if we summarize everything that we have seen in this video so far, what we have to take care of with any powerful platform for ADC is the homogeneity to begin with. Drug to antibody ratio, can that be regulated? What sort of chemistry can be offered? And will there be opportunities to have a regulation there? Will there be an opportunity to control the residue on which the toxins are conjugated? Can we control the bond strengths? What will be the flexibility with the linkers that we want to choose? And can we also control the site of conjugation? Now, we are looking into the LDM platform for addressing some of these challenges, to be precise. So let me give you one representative example that we developed for LDM that is used for histidine modification. So the monoclonal antibody of our choice was trastuzumab in this case, and we also selected M10 or DM1 as the toxin. Now, the basis of this choice was to make sure that we can make a head-to-head comparison with Kadcyla, which is the FDA-approved drug from Roche. And what we were excited to see is that the LDM-derived ADC does a pretty great job in inhibiting the HER2-positive cells. And it does perform slightly better in comparison to Kadcyla in every trial that we did. Apart from that, it was important to see that while it inhibits HER2-positive cells, it really had no negative impact on the HER2-negative cells. So it was quite specific towards the HER2-positive cells in that regard. Now, as we discussed, in this case, the site of conjugation of drug is histidine, and we can tune that to a great extent. But apart from that, The LDM platform also provides a lot of flexibility in terms of the residue of conjugation. So we can take this forward, and we can even apply it for precise and modular modification of the lysine residues. And this method works again with a diverse set of proteins, but then we can very conveniently extend it for the synthesis of antibody-drug conjugates. So let me show you one representative example from this category, where we made a ADC where the toxins were conjugated specifically at lysine 169 and lysine 395. And this is the bar that represents the inhibition from LDM derived ADC in comparison to Kadcyla. What you would notice is that it not only does much better than Kadcyla, it's almost equivalent to what a free DM1 is doing. And that's really remarkable, because not only it can provide inhibition to such a high level for HER2 positive cells, it really does not inhibit HER2 negative cells at all. So this, I hope I was able to convince you that chemoselectivity, site selectivity, residue specificity, and modularity provides a pretty comprehensive technological platform for precision engineering of proteins. And for last 1 year or so, some of our group members are looking into how we can translate that into the live cells. And I'll be glad to discuss about some of those stories at the next occasion. But we are really excited about what will happen when we are going to translate them further to the animal models and beyond. Now, all the technologies that I discussed today and a few more are accessible to the scientific community through Playable Tech. And with that, it's time to thank my group members who did all the real work in the laboratory. We are also very thankful to all our collaborators who make immense contribution in this multidisciplinary research field. We are very thankful to all the funding agencies for their generous support over the past several years. And with that, thank you all for your kind patience. I'll be glad to take your questions.